Very good morning, students. So today we are starting with another topic in Sycon, that is the reproduction. So first of all, you can go with the previous video. A description has been added with a link. So first of all, you should go with the process of fertilization in that particular video, and then you should start with this topic, the reproduction in Sycon. Now, here, after fertilization, the zygote was formed, and that zygote was lying in the mesenchyme of the sponge. Now, in this part of the video, we are going further after the formation of the zygote. So, we start with the development of the Sycon. So, the development of the Sycon can be divided into three periods. So, the very first period is the early embryonic period. And this embryonic period can be divided into two further phases. So the first phase is the cleavage. So cleavage is the breaking down of the zygote. So here the zygote, it is subjected to three vertical divisions. So here you can see a zygote. It undergoes three vertical divisions to form 8 blastomeres. Now a horizontal division occurs, which divides the 8 blastomeres into 16 blastomeres that are in two tiers. So the 8 micromeres which are present in the upper tier, they are small in size, and the lower 8 cells are larger in size. So this division results in the formation of 16 cells. Now after that, the upper 8 cells, they divide mitotically and they increase in number, and they bear flagella. So in the lower diagram, here you can see the formation of a blastula. So in this particular structure, we have got a large number of micromeres which are violet in color, and we have got 8 lower macromeres which are yellow in color. So here you may find the micromeres are flagellated, whereas the larger macromeres are non-flagellated. This blastula is sometimes referred to as stomoblastula because it has got a mouth, the opening, so it is hollow from inside. So that's why it is called as stomoblastula. And this is our second phase, the stomoblastula, as the first one was the cleavage. So now we have stomoblastula formed in the mesoglia. So everything is occurring in the central layer of the Sycon. Now after the formation of stomoblastula, the second period starts, which is a larval period, and it is further divided into two phases. The first phase is amphiblastula formation, and the second is gastrula formation. So now we are starting with the first phase, amphiblastula formation. So in this, what happens, the stomoblastula, it undergoes inversion process. So here in this process, whatever is inside the blastula, it comes towards the outside. So the flagellated cells, they are inverted, and the flagella, they are coming towards the outside. So here you can see the flagella are towards the outside. Now this particular larval form is called as amphiblastula. Amphi means both. So it has got both types of cells. Half are flagellated, and we have got non-flagellated cells also. So that's why it is amphiblastula. So here you can see the amphiblastula from the outside. Up to this level, the whole of the development is within the sponge. It is within the mesenchyme of the sponge. Now this amphiblastula has got flagella on the outside, and now it can swim. So it comes out of the body of the Sycon through the osculum. So in this diagram you can find out the formation of the larva inside the body of the sponge. Now this amphiblastula, it is going out in the spongocoel, the central body cavity. Now it is leaving the osculum of the Sycon and it goes out, and now it is a free living larva. So this is amphiblastula. Now this free amphiblastula, it enters into the second phase of larval period, which is the gastrula formation. Now what happens, this amphiblastula, it moves freely out in the water, and at last it settles down and it attaches to the substratum. So this attachment, it provides a stimulus for the gastrula formation. So gastrulation occurs. So here in gastrulation, what happens? The macromeres, now they divide rapidly and they cover the micromeres. Now here in this diagram you can see the macromeres have covered the micromeres, and the flagella, they are again coming towards the inside. So this process is called as gastrulation. Now after gastrulation, the third period of development, which is the post-larval period, it starts. So this period is also called as metamorphosis. Now here what happens, the larva, it fixes, it forms a gastrula, then it lengthens. So it becomes long, cylinder-like structure, and it forms an osculum. So here an osculum is formed. Ostia are also developed. Now just see the gastrula. It has got the macromeres towards the outside and micromeres, the flagellated cells, towards the inside. So what happens, the macromeres which are towards the outside, they develop the pinacoderm, and the micromeres which are towards the inside, they form the innermost layer of the Sycon, which is the choanoderm. So thus a stage is formed which is called as Olynthus stage. Now after this stage, radial canals are formed and there is further differentiation, and finally we get a full Sycon. So that was the development of the Sycon.